little bit more about the science part, and what's bad is that I'm not the scientist, so I will cover just in very broad terms, and every question should be addressed to the real scientific people that I will quote at the end of the talk. So Vitality is a high-performance computing center, what is important is that its strategic goal is not only to provide hardware and storage to our users, but to provide the key competencies that so that they are able to solve their problem. Because our main users are biologists who don't always have the competencies to solve their problems. So, as Konstantin mentioned, these people cannot just be thrown at a HPC and find a way by themselves to solve their problem. They need a whole set of competencies that we try to address within our center. So, Vitality is a coordinated resource, so it has a part of infrastructure. Okay. So the infrastructure is known to you by my talks that you have to support every time that you attend to a hpc-ch conference. But what is important is that we also provide expertise about bioinformatics software. We also provide database design and algorithmic design to our users. It is very well integrated with the Swiss Prot knowledge by curation, because sometimes you solve a problem, but then you have to comment, add information to this scientific knowledge you have gathered, so that others can also use it. And the biocuration is a very important part of the whole services that we are able to provide. We have a very tight connection with education groups, because as part of the university we organize courses in different fields of bioinformatics, and we have a very good tie with research groups by the fact that there are bioinformaticians that are connected to SIB that are part of the research groups and the core facilities. Vitality is connected to the different universities, to SIB, first of all, because it's a group of Swiss Institute of Bioinformatics, but its partners are the University of Lausanne, EPFL, University of Geneva, University of Basel and Freiburg, and we collaborate with them to provide them competencies and support for their biology-related projects. We also have very strong ties with the platforms, which are facilities where biologists can bring their samples so that they are sequenced and pre-analyzed. So it is the workflow is that a biologist, which is an external user, goes to a facility, it gets the samples to be processed, then it's pre-analyzed, then it might or might not require services from Vitality to gather knowledge about the biological sample which was provided, and maybe we have to set up a website so that they can share their information. So this is more or less the workflow that we provide in Vitality. So as you know, it's a distributed competency center, but yet centrally managed. It means that we presently manage 3 clusters. One, the original one was built in Lausanne 10 years ago. So sometimes later this year you might get an invitation for an anniversary. There is one in EPFL, one in Geneva, which total more than 3,000 cores. We also provide these entities large-scale data archive as form as an HSM. We also can provide CPU, GPU based computing. It's structured like this, so there is a team that is made out of the director Ionask Scenarios, senior scientists who manage projects and collaborations, bioinformatician, postdoc, PhD students, and system administration, and these have a direct connection to the user, who are either the embedded bioinformaticians, which are in direct contact with lab biologists, university core service platforms, or advanced users. But we also have a lot of novice users that we have to support, mainly via the use of web interfaces. So, to support, to provide these services, we have 3 clusters. One external cluster where we mirror tools and databases with around 2.7 petabyte of nearline storage. We maintain also more than 300 bioinformatics packages, which make more than 1,000 binaries, which go in all fields of bioinformatics like phylogeny, genome analysis, proteomics, and population genetics, and so on. And we also update a complete set of biological databases that we download every time that there is a new version, and we update these databases on all 3 clusters. This is quite a huge task, and therefore the Vitality group now is made out of 44 staff members, 5 sysadmins, no 4 now, but 400 users, active users per year, and we are involved in 165 projects which encompass all fields of bioinformatics. In 2012, we ran more than 20 million jobs for 678 CPU years. Currently, we have a new data ingress of more than 25 terabytes a week. So data management is a big issue for us. So these are the people that can help our users with different problematics. So Christian Isley and myself take care of the HPC. Heinz Stockinger is a representative for all supercomputing and grid problematics. Oncogenomics and personalized genomes are projects are managed by Brian Stevenson. We have experts in metagenomics, metabolics models, evidence-based biomedicine, and computational systems biology. So these are the people that manage the high-level projects and who can address the user to the right group, to the right, and develop with them the solution for their problem. So these are some examples of R&D enabled through Vitality. So our users have been quite successful because in the last 5 years they could manage to publish 28 papers in high-profile newspapers. but we are part of the EMEDIA consortium, which is a private-public partnership European project. We provide the food and health and agriculture organization epidemiological surveillance database of influenza virus through a website. Vitality have developed a new algorithm to allow the detection of trisomy and chromosomal abnormalities by using next generation sequencing in the blood of the pregnant mother. And it has developed a novel hierarchical density-based clustering algorithm, which is mostly used in flow cytometry data, but it can also be used in satellite image analysis or EEG. So I will try to describe the scientific part, well, so in broad terms, the scientific part of the what has been done. So EMEDIA is a consortium for diabetes research. It's a collaboration between a lot of academic and pharmaceutical research institution with a collaboration all over Europe. And so the idea is that in vitro models are studied as well as so these in vitro are cells coming from samples which are connected to a repository of human beta cells. This is there are groups that make beta cell imaging, and here we have in vivo models, and SIB does the system biology of and coordinates the knowledge of all these groups collaborating together. So Vitality provides the data storage and sharing via one database, and the group led by Mark Eberson develops web-based data mining tools so that in order to find novel biomarker identification in diabetes. So we also host a database for human and animal influenza viruses. So it's presented as a web interface that collects on one side epidemiology information and also genetics, and which allows to map geographical distances to genetic distances, and to be able to do a correlation so that FAO with their Empress can have a link to the from the outbreak date and location to the matching various sequences. Now, non-invasive prenatal diagnostic of chromosomal anomalies. So you can have DNA, free flow DNA in the blood. In some cases, some cancer patients or patients with inflammatory diseases or pregnant women have a DNA, free DNA in their blood. And you can have fetal DNA in the plasma of the mother, of the pregnant mother. which is detectable from the 7th week of the gestation. This DNA disappears just after the birth, so it's sure that it's a fetal DNA. So the test that has been developed allows to detect anomalies on the chromosome 21. So these are trisomies as well as chromosome 18 and other chromosome. And in a double-blind experiment, the test that has been developed by Vitality has been very successful because it detected almost but one trisomy or chromosome aneuploidy or triploidy. And it's very specific because it doesn't cause any false positive. So it's a very important test which is now sold by a company called Medisupport. The sequencing is done by a company in Geneva called Fasteris. And this has been tested with the help of the King's College in London. And the authors of this test are Nicolas Gay, Christian Risley, and Ioannis Xenarios.
Can I say something?
Yeah.
So basically, I think Roberto was very, very modest about this. But I think what this means is whenever you have your wife being pregnant recently, coming up in the next whatever years, you can do a test, because everybody who's ever done an amniocentesis test, which is the invasive test where you get a needle into the uterus of the mother and then take out some pieces of the DNA and analyze it or just look at it, it's usually a very high risk, at least a 1% risk that the baby dies. That's a test, does a similar thing, but basically at no risk because it takes blood from the mother. And this is, I think it's a very important breakthrough that we've developed. And I'd say it a bit more unmodest than Roberto did, especially when you're affected. Once you have your wife doing an amniocentesis test, and you have to think about, shall I do it? Shall I do the test? Shall I risk the life of my baby? Then you probably think twice about coming. End of marketing.
Thank you for the auto-congratulation.
I was not part of it, so I can just comment.
So Vitality has also developed a test for flow cytometry measure. So these are cells that are marked with a fluorescent marker in order to detect some proteins of the membrane. So you can attach different markers for different membrane proteins, and then the cell sample is flown through a cell where it's illuminated by different lasers, and then you measure the luminosity and the response to every marker. And in the end, you have some kind of blob of different colors, and you would like to understand what marker attached to what cell. And it can be very complex because you have a lot of a big number of individuals. You want to test many doses over a time course and with different markers, and you have numbers of cells in the million range, and it can do millions of points to cluster. And this algorithm allows you to find a cluster of significant importance in very complex multidimensionality vector samples. So, in conclusion, Vitality has been shown to be a very important asset. We help a lot of groups solving their problems and gather information from their data. What is important is to have people that have good bioinformatics competencies attached to an HPC and high throughput computing infrastructure. These people must be very closely linked. So we should be, one of the successes is that we have storage and archival for the projects. And we have a long-term maintenance of life science and data. And we have a tight integration with diverse technology platforms, from genomics to proteomics and so on. Because usually what we notice is that moving data from one facility to another takes too much time, and it's important to have these things attached close to one another. And it's also best for the users that they know that they can go to someone to ask him, maybe because in these research projects it's sometimes difficult to set up a very complex design. People prefer to discuss and to explore the solution together with the bioinformatician and to develop their workflow for their solution. So all these slides, of course, I was not the author. It's these were the authors of these slides, and with that I am finished. So.